Hi guys, my name is Natalie Quinteros and I'm a bio major at Oglethorpe University. So to start off, I wanted to give an introduction to why I chose de-extinction. So it was mostly following on the idea that I did some research focusing on the ethicality of de-extinction based on a point of view of a doctor. Based on that, I became a little biased towards the topic because became interesting to me how of following the ideas of Jurassic World, Jurassic Park, and if that's really going to happen. So not only that, the technology and the growth in this in genetics that leads to this topic is really strong and led me to just want to do more research on the mythology, what led them to pick the specific species, and what could be the newer species upcoming. So to start off, I wanted to do I just wanted to give a definition of de-extinction. So this definition was given by the International Union for the Conservation of Nature. It just states that de-extinction is the ecological replacement of an extinct species by the alternation of phenotypes through different techniques. These techniques can be the artificial selection, the backbreeding, precise hybridization done through genetic modification. And these are all techniques that we're going to be going over today during our presentation. So moving on to the introduction of our topic. I know that every time you think of de-extinction, you think of the T-Rex eating that big ball and trying to attack everybody in Jurassic World. So you know how at the end of the movie, everybody ends and mostly the park was done for monetary beneficials and everybody's trying to get money out of it. De-extinction is a little bit more than that. And it takes a lot of time for this research to be done. It's very tedious and it's mostly for our environment. So de-extinction is just a process that would restore various of our ecological functions that would allow the sustainability of our ecosystem and just allowed it to increase, have more biodiversity and abundance. So this is more for the beneficial of us and our earth than like monetary, and then like having a park just to go see the animals, more like having these animals introduced to the wild and grow and have our ecosystem thrive. So I kind of did an outline of the types of techniques we're gonna be going over, and I wanted to go through some going more into details of these techniques, just so that we understand what it's going through and the processes that these researchers are taking on. So we have artificial selection. I wanted to go over the definition of it, and it's just the identification of the desirable trait and the organism that you wanted to, and the steps taken into enhancing those, and see to be able to perpetuate those into the future or generations. So a type of artificial selection is backbreeding. Backbreeding is just a form of artificial selection in which you consist of the reproduction of an organism. An example of this that we see in our everyday life is just dog breeding. So I picked an example of dog breeding that included a pure Labrador, and then we have a pure poodle that then gives us Labradoodles. Then this is something that you can see every day. You go to any park and you ask someone what type of breed their dog is. They can either be pure or they can be a mixture of those. Then we have precise hybridization. Precise hybridization is just the process of combining two complementary single either DNA or RNA, and then having those form a single double-stranded molecule through base pairing. Then we move on to just the de-extinction research through breeding, in which it focused on the Phacloperegrinus natum. This species, mostly this research was done by Harrison B. Tordoff and Patrick Redick. These were the lead researchers for this experiment specifically that was done at the University of Minnesota around the 2000s, in which we see the Falco peregrinus being the species they focus on, and then it went extinct in the wild in the 1960s, and it had to be put into captive breeding. Captive breeding just means that they put all the species almost extinct into a small area. It could be like into a zoo or in a way, in a controlled environment that allows them to being able to grow and survive in a safer way. Thanks to the FB Peregrinos Anatom Recovery Program, they were able to reintegrate around the late 1970s, in which they had a genetic mixture of the anatom with the Quinteros, the mutant, and the polio. And they had this genetic mixture that allowed the environment to select the most successful genotypes and phenotypes that later led to the FB anatom to adapt to their new environment and migrate east and Midwest, in which 5 out of the 7 subspecies that were released were making a new contribution to the new population. So they thought that this experiment was successful and had a positive result that now, well, back then in the 2000s when they did this research, they thought of continuing doing the same experiment with different species so that they can allow them to have a chance to thrive if they're close to extinction. So we move to a more technological and genetic process of de-extinction that includes these concepts. I wanted to go through these concepts in like specific and just go through some definitions so we understand how this concepts come into play with the de-extinction and the process of genetic modification. So we have in silico. In silico means the experimentation done through a computer. This phase is really important. It's usually put into phase 1 for any research doing genetic modifications due to it being to, as they have the genome of distinct species, or they're putting together that genome and they compare it to the subspecies genomes or the model organism. They try to get those desirable traits to compare those and put together the new genome of the species that they're trying to bring back. Then we have the in vitro. In vitro is just something done in the lab or outside the living organism. Then we have in vivo. In vivo is just a process that takes place inside the living organism. Then we have the ex situ, which is outside their natural habitat. And then we have in situ, which is the experimentation in the original habitat. And to begin with, we had the pathogen pigeon, which is a process of de-extinction via genetic modification. I wanted to go back to the precise hybridization process concept so that we can see how this come into place and these concepts are really important to it and how this circle just goes on and on in the phase 1, in which the passenger pigeon research took place at the University of California, and it was led by Benjamin Novak, which with the help of a lot of fellow researchers, advisors, and partners that did separate researches to put all this together, in which they had the genomes of the template species sequenced from a wild individual that was in situ. Then they had a captive individual that was ex situ, and then they had an embryonic fibroblast cultures that was in vitro. And then they were able to run this through programs to identify the candidates that share alleles with the pathogen pigeon that led us to the in situ. And you can see how this circle just goes back and forth until they find their model organism. That takes us back to the phases of the passenger pigeon research specifically. So the phase 1 just takes up the genome research that began in 2012, where they began to sequence the DNA from the passenger pigeons. This helped them see that the passenger pigeon was a suitable candidate for the experiment itself for de-extinction. that then led them to the phase 2 of the revival, in which it began around 2017, where they had their first experiments. They determined Bantel pigeon was their model organism. They started editing genomes of living birds to the extinct passenger pigeon. So this is the phase that they're currently on, and they're still working on to have the complete genome of the passenger pigeon. So far, they haven't had the complete genome, but they're still working on it. Then we have the phase 3. This is the future one that we hope to see sometime soon. This is around, hope to start around 2024, in which they have the restoration. They have been working on the restoration research for about starting in 2017, when they had the revival, in which they put together research by the Bronze Zoo, which is one of their partners, that they did a captive breeding research on other pigeons to see how they could raise the flock for the passenger pigeon when they are able to bring it back, in which lead us to the timeline of the passenger pigeon de-extinction. In 2012, as we see here, we have the phase 1 started, and then we have the passenger pigeon genomics and the Bantel pigeon genomics, and then they start comparing those two to see what traits we can bring out from the Bantel that share with the passenger pigeon, just to start putting that whole genome together. And now we have the genome editing research here, and then hopefully we have the de-extinction by the end of 2024. That then would lead us to the phase 3, which we have the captive breeding, where they're put in a controlled environment like a zoo, just have them grow, reproduce, keep growing in population to be later on softly released. This would then lead to the wild release, which is not under a controlled environment, and they just to let them thrive into this new environment. As you can see in this timeline, de-extinction takes time and it's very tedious work. It takes a lot of computer research that then leads to a lot of hands-on in the lab, and then observative research. So it's research that's taken out from different partners and different people conducting a series of experiments. So it's my respects to the researchers following this and how long they have been working on it with no results so far, and hope to see some results in the future. So then we lead to the Mammuthus primogens. This is a very similar, follows the passenger pigeon through genetic modification. So this one's a little bit more known. I tend to hear more about this one due to the idea of the woolly mammoth and the Ice Age. So as we know, this species went extinct in 1650. So now this project is being led on by George Church. It's conducted at the Harvard University at the Wyss Institute. It's also promoted by the Colossal organization. This organization focuses on de-extinction and bringing back and providing an environment for this species to come back onto and being able to thrive. So this organization, George Church, is not only one of the researchers behind the de-extinction of the woolly mammoth, but is also one of the co-founders of the Colossal and one of the CEOs over there that leads the project. So following on more details about the project itself, we have the project model being the Asian elephant, being one of the most closest related subspecies to the woolly mammoth that would let them be, that led to the African elephant being the surrogate for this project. As with that being said, a hybrid Asian woolly mammoth embryo is going to be implanted into a healthy African elephant surrogate. They thought that the African elephant surrogate would be a more suitable, being able to, just more suitable to be able to carry this elephant due to the fact of its mass and how the woolly mammoth tends to be a little bit larger. So, and just the traits that they're putting together. So I wanted to highlight a big difference between the passenger pigeon research and the woolly mammoth research. So the way that they're able to, the passenger pigeon takes a little bit more time based on the genetic and the genome edited research and the computer base. And mostly the phase 1 of in silico has taken so long due to the fact that they don't have the full genome of the passenger pigeon. On the other hand, the woolly mammoth is more of a, they're able to get their DNA through a more conserved DNA through their bones due to they stay more conserved because they were frozen and the place that they went extinct. So the fact that the bones of the woolly mammoth was frozen allowed it to be more conserved and extract DNA easily than compared to the one of the bones that they got from the passenger pigeons that stayed in a zoo that was dusted, and it was in different environments and circumstances. So far what they have been able to do with the woolly mammoth is that they have been running through the CRISP system, the replacement of the 14 loci, and the elephant genome, and with the version of the woolly mammoth. So they've been able to successfully do that. And they've been able to successfully edit the living elephant cells, which is a great advancement in technology and in genetics. So far for this project, we don't have a date where they expect to have some results, but they have shared their enthusiasm and how they're close to comparing the genomes and putting together a whole genome of the woolly mammoth and putting the embryonic surrogate into, putting the embryonic into the surrogate and hopefully have some results soon. That leads us to the take-home message for today, in which we have the de-extinctions of research development that is part of our present, our future throughout different techniques. Not only that, de-extinction it's a topic that grows every day, and that leads us to just the growth in technology and the new ideas that are coming as to if the CRISP system is results to be successful and the extinction through woolly mammoth is complete, then it could then lead to cloning, which is a new advancement in technology. And we see the changes of technology and genetics. It has come from backbreeding, which is artificial selection, that then led to genetic modification through the hybridization process, in which we see the woolly mammoth, the passenger pigeon, and the dodo bird coming to life. So I hope to hear some more results as to de-extinction keeps growing every day and will probably not only affect us and our generation, but also probably our future generations. So that'll be all. Thank you.